Dear learners, now let us conclude. In this week, we shall got acquainted with enzymes, protease inhibitors and amylase inhibitors. We studied that enzymes are biological catalysts. They increase the rate of chemical reactions taking place within living cells without themselves suffering any overall change. The reactants of enzyme catalyzed reactions are termed substrates, and each enzyme is quite specific in character, acting on a particular substrate or substrates to produce a particular product or products. Their activity may be inhibited by compounds called enzyme inhibitors. They are substances which alter the catalytic action of enzyme and consequently slow down or in some cases stop catalysis. In this module, we have studied in detail three common types of enzyme inhibition, namely competitive, non-competitive and substrate inhibition. Under amylase inhibitor module, we have seen that inhibition of alpha amylase, an enzyme that plays a role in digestion of starch and glycogen, is considered a strategy for the treatment of disorders in carbohydrate uptake such as diabetes and obesity, as well as dental caries and periodontal diseases. Plants are an important source of chemical constituents with potential for inhibition of alpha amylase and can be used as therapeutic or functional food sources. Amongst the phytoconstituents that have been investigated, flavonoids are one of them that demonstrated the highest inhibitory activities with the potential of inhibition related to number of hydroxyl groups in the molecule of the compound. In the module on protease inhibitors, we learned that protease inhibitors are molecules that block the activity of proteases. Protease inhibitors can either be in the form of proteins, peptides or small molecules. The first plant protease inhibitor was isolated from soybeans and characterized in 1947. They are grouped in two inhibitor families and are represented by two predominant inhibitor from soybeans, the Kunitz soybean trypsin inhibitor, that is STI, and the Bowman-Burke trypsin and chymotrypsin inhibitor, that is BBI. A trypsin inhibitor is a protein and a type of serine protease inhibitor known as serpin that reduces the biological activity of trypsin by controlling the activation and catalytic reactions of proteins. Protease inhibitors have been examined extensively as therapeutic agents, primarily to deal with various human cancers, effective against cardiovascular diseases, osteoporosis, inflammatory diseases and neurological disorders. Thus, we have learned enzymes, protease inhibitors and amylase inhibitors. In the next week, we shall study active biodynamic principles in spices. Thank you and happy learning experience.